Okay, so welcome to this next video in the playlist on cis-loop ligand-gated ion channels. Okay, so in this video what we're going to talk about is two very recently discovered channels known as ELIC and GLIC. And basically, ELIC and GLIC are ligand-gated ion channels. Well, actually, ELIC is a ligand-gated ion channel. GLIC is actually pH-gated, so it's controlled by proton concentration. Okay, and these two channels are very, very similar in structure to cis-loop ligand-gated ion channels, and we've been able to use them to study the opening properties of the cis-loop ligand-gated ion channel. So that's why they're very, very important. Now, they are both found in bacteria. ELIC is found in a bacterium known as Erwinia chrysanthemi, and GLIC is found in a bacterium known as Glioblastoma violaceus. Okay, but I'll write down their names later on. At the moment, what I want to talk to you about is the cis-loop ligand-gated ion channels, and then what we'll see is how ELIC and GLIC have a very similar structure. They have slightly modified structures, but we'll talk about that when we come to it. Okay, right, so we'll start off with cis-loop ligand-gated ion channels, the type which are found in humans. Okay, so cis-loop ligand-gated ion channels, and ligand-gated ion channels is often abbreviated to LGIC like that. Okay, so, right, and ELIC and GLIC aren't found in humans, they're found only in their bacteria. Okay, right, so cis-loop ligand-gated ion channels then. So, the cis-loop ligand-gated ion channels, examples of them, the key examples in humans are the nicotinic acetylcholine receptors, okay, the Gaba receptors, which are the form of Gaba receptors which are ligand-gated ion channels. The Gaba B receptors are G protein coupled. Gaba is the major inhibitory neurotransmitter in the brain. Then we also have the glycine receptors. Okay, so GABA is the major inhibitory neurotransmitter in the brain. Glycine is the major inhibitory neurotransmitter in the spinal cord. So again, we need ligand-gated ion channels that respond to glycine, and those are found in the spinal cord, and they are again cis-loop ligand-gated ion channels. And finally, the other example is a receptor to 5-HT, 5-hydroxytryptamine. Okay, also known as serotonin, and this is the type 3 serotonin receptors. Okay, so you might want to put an R at the end to show that it's a receptor. So there are many different types of serotonin receptors, most of which are G protein coupled. So for instance, the 5-HT1 receptors, the 5-HT2 receptors, they're both G protein coupled, but then 5-HT3 is one of these cis-loop ligand-gated ion channels. And this family of 5-HT receptors are the only known receptors for serotonin that are ligand-gated ion channels. Okay, right, so let's have a look at the structure of these types of receptors, because that's what we characterize them by, why they're all grouped together. Okay, and if you want some examples of ligand-gated ion channels that aren't cis-loop ligand-gated ion channels, so some non-examples, then these glutamate receptors will be non-examples. So for instance, the AMPA glutamate receptor, the NMDA glutamate receptor, the kainate receptor, those are all ligand-gated ion channels, but they are not cis-loop ligand-gated ion channels. Other examples are more niche. So for instance, the P2X receptor, the purinergic receptor for ATP, that's a ligand-gated ion channel which isn't a cis-loop ligand-gated ion channel or a glutamate receptor. So these are two non-examples, the P2X receptor as well. Okay, so now let's talk about the structure of cis-loop ligand-gated ion channels. So, their structure looks something like this, or at least this is how I'll draw it for a cartoon purpose. Okay, so here is our channel going through the membrane. And I suppose I should now, since we've discussed this in our previous video, I should add the intracellular domain on here. So I'll put an intracellular domain at the bottom there. Okay, right, so you have basically this protein is made up of 5 subunits. 1, 2, 3, 4, 5, like that. Okay, and each of these intracellular portions down here, making up the intracellular domain, is also made up by these subunits. So, let me take one of these subunits out, one of these protein subunits that makes up a fifth of this ligand-gated ion channel, and let's have a look at its general structure, okay? Right, so the general structure of one of these subunits looks like so. Okay, so if this is the phospholipid bilayer shown here, then here's the amino terminus of our protein. Okay, then you have something known as the cis loop here, which is a loop in the polypeptide held together by disulfide bonds. Then it traverses the membrane once, it traverses the membrane a second time, a third time, and then a fourth time. Okay, and then here is the C-terminus of the protein. Right, and actually I just want to amplify one thing. I want to make this loop here bigger. So let me just modify that. So I'll put some colour on to repair the damage that I've done there. Okay, and this loop here is known as the intracellular loop, and that's the portion responsible for this intracellular domain down here, as it's called. Okay, right. So this is the intracellular loop, or it's also known as the M3-M4 loop. So this is the intracellular loop. Okay, or also known as the M3-M4 loop. Right, okay, so basically what happens is each one of these protein subunits, it has this transmembrane portion here made up of the M1, M2, M3, M4 membrane spanning alpha helices, and then this intracellular domain is made up of this intracellular loop, and every single one of the 5 subunits provides an intracellular loop into this intracellular domain. So the structure ends up looking more like this if I draw it here. Okay, so if I draw the protein again, like so, then you have these 5 subunits making up the pentamer. Okay, like so, 1, 2, 3, 4, 5. And each subunit basically has this intracellular loop coming down. Okay, and what happens is all of the intracellular loops from all of the subunits come together right at the bottom. Okay, so all 5 of them come together and sort of form a connection at the bottom. And as you can see, between these neighbouring intracellular loops, so here's one intracellular loop, here's another intracellular loop, here's another intracellular loop. Between these neighbouring intracellular loops, there is a gap known as a porthole, okay? So this is called a, is that right? A porthole. I think it's called a porthole, okay? This is a portal. That's what I got wrong. A porthole is what I was saying. A portal is what it's called. Sorry. I was thinking a porthole. That's something in ships. No, portal. Something which you go through. Right, okay. So, what this ensures is that when the ions come in through the pore, they then are directed off. And their direction of motion when they come through these portals will be parallel to the membrane. Whereas if you didn't have this intracellular domain, as it's called, then when the ion channel, when the ions come through the pore, they go zooming off into the cytoplasm. So you want to direct them towards the inner leaflet of the phospholipid bilayer, so that the change in membrane potential that you get across the cell membrane in response to the ions coming in is maximized. Okay, so that's the function of the intracellular domain. Right, so let's talk a little bit more about this cis loop here and what that cis loop actually is. So, a cis loop then, basically a cis loop is a loop in the protein that is held together by a disulfide bond between 2 cysteine residues. So let's have a cysteine in one of these polypeptides of the loop. So here comes the cysteine. So here is the amino terminus, here's the alpha carbon, here's the R group of the cysteine amino acid. with what would be a thiol group, but the thiol group is going to be involved in a disulfide bond, so the hydrogens come off. Okay, and then the polypeptide comes round like so, and here's another cysteine over here. So here's the alpha carbon, and here's the R group of the cysteine, so here's the methylene group, and it too has a sulfur coming off, which would have a hydrogen on if this was a normal cysteine, but they have bonded together, so they've lost the hydrogens. The two hydrogens will go off together. And this new structure that you've got here, let me just complete now this cysteine. This bond here between these two cysteine residues is known as a disulfide bond, or also called a disulfide bridge. So what you've got is this loop in the polypeptide structure held together by a disulfide bond between two cysteine residues in the polypeptides, and this is what's known as a cis loop. And it's because CYS is the 3-letter amino acid code for the amino acid cysteine, which is the amino acids involved in this disulfide bond. So cysteine is also known as cis, or its single-letter amino acid code is just C. Right. Okay, so these subunits that make up the cis loop ligand-gated ion channel, they have this cis loop, they have these 4 membrane-spanning domains, and they have this large intracellular loop, or M3-M4 loop, which makes up the intracellular domains here. Okay, right, so that's the basic structure of a cis loop ligand-gated ion channel. Now let's have a look at ELIC and GLIC, but we'll do that in the next video.